you for the introduction, and thank you for the organisers for giving us the opportunity to talk. I'll be presenting what's really very top-line data of a study of AVXL 273 or blarcamesine, a randomised double-blind placebo-controlled multi-centre phase 2b/3 study in patients with early Alzheimer's disease. My disclosures are listed. In addition to my disclosures, I'd like to add a disclaimer. If you thought Roche did very well getting their data to present top-line results yesterday within 3 weeks with a massive team of statisticians behind them, we had database lock on this study 2 weeks ago today. There's been a much smaller team of biostatisticians analysing the data during that time, and it finally reached AVXL yesterday, and I received the final slide set for this presentation within the last hour. So my apologies if my presentation isn't as slick as it might otherwise be. It is good, however, to highlight results of a positive trial of a study of a novel platform approach to address the complexity of CNS diseases by activation of the sigma-1 receptor by an orally available small molecule AVXL 273 or blarcamesine. And it's my pleasure to announce that the study met its primary endpoints, both of them, as well as its secondary endpoints. So we showed a statistically significant reduction in clinical decline in global clinical study of this global clinical study of patients with early Alzheimer's disease. We'll be presenting the rationale for the use of the molecule in this population, and flag that we've also got human patient data and a number of other neurodegenerative and CNS diseases, including Rett syndrome and Parkinson's disease. So get the money slide out of the way first. AVXL 273 treatment over a period of 48 weeks compared to placebo slowed the decline of cognition and function in patients with early Alzheimer's disease. Of course, in any trial, you're going to get patients who are going to improve both cognitively and functionally in both treatment and placebo arms. In this study, if you did improve cognitively in the study, you were 84% more likely to have received active drug rather than placebo. And it slowed cognitive decline compared to placebo at 48 weeks by about 45%, so a significant cognitive slowing using the ADAS-Cog. There was also a 167% greater likelihood of showing improvement in your ADLs if you were assigned to the active treatment arms of this study as well, with a clinically significant score difference of 3.5 points or greater between active drug and placebo groups. And in terms of the decline in cognition measured by the CDR sum of boxes, progression was slowed by 27% compared to placebo over that time period as well. And in addition, the drug was safe and well tolerated. So in terms of the mechanism of action of the drug, the sigma-1 receptors largely unknown to many people, but we've heard from earlier in the conference that Alzheimer's disease pathology is certainly very complex. It's more complex than beta amyloid and tau. It involves a number of other cellular disruptors, including mitochondrial dysfunction, inflammation, synaptic dysfunction, and cellular stress combined with protein misfolding. A variety of targets present themselves, obviously, because of this complexity, and a biological modulator of all of these downstream pathways is actually required. And this is where AVXL 273 comes in with its activation of the sigma-1 receptor. In many ways, it targets all of these downstream pathological mechanisms that are known to be implicated in Alzheimer's disease. And a simple way of understanding it is that it's a bit of a rescue receptor in many ways. When a cell is functioning normally, the sigma-1 receptor is largely inactive, but when abnormal proteins and other cellular homeostasis mechanisms are thrown out by neurodegenerative disease processes, activation of the sigma-1 will act to remove abnormally folded proteins from the cell. So in many ways, it's agnostic as to whether amyloid, tau, or some other process is involved. It's a significant upstream modulator of all of these pathological processes. So sigma-1 activation has been shown to modulate multiple aspects of neurodegenerative processes in a variety of animal, human, and disease models, some of which are listed here. And it's also an important membrane protein in terms of regulating cellular homeostasis. Impaired sigma-1 function leads to dysfunction in endoplasmic reticulum mitochondrial crosstalk, calcium homeostasis impairment, and endoplasmic reticulum stress response. So the sigma-1 receptor could have a therapeutic role in a number of neurodegenerative diseases, and indeed the compound's being trialled in a variety of other conditions in addition to Alzheimer's, as I've mentioned. The study design, 509 patients, they're an early Alzheimer's disease population who had confirmed Alzheimer's disease pathology. Patients were aged between 60 and 85 years and had an MMSE score between 20 and 28. Patients were randomised 1:1:1 across 3 arms. There was a placebo arm, a 30 milligram dose of AVXL 273 and a 50 milligram dosage arm. The primary endpoints were the ADAS-Cog, the ADCS-ADL, and a key secondary endpoint being the CDR sum of boxes. I won't be presenting all the data today, largely because it's not been analysed. These are very top-line results, but there are a number of pre-specified analyses that will be conducted to confirm observations from an earlier phase 2A study, which showed that possession of the sigma-1 wild-type gene, which is common to about 80% of the population, denoted an extra degree of response when treated with this compound compared to the mutant variant of the gene. Those data, along with other analyses, will be presented in due course. So our top-line data in relation to this study, the baseline characteristics of the patient population are presented here. You can see that they're relatively well matched across all 3 treatment arms of the study, both age, gender, race, use of Alzheimer's disease medication, MMSE and ADAS-Cog scores, and their ADCS-ADL scores. They're also reasonably well matched across the CDR sum of boxes at baseline. So these are our primary endpoints, which I've mentioned, along with the key secondary endpoint. The top-line data in relation to efficacy are as follows. If you did improve cognitively during the study, you were 84% more likely to improve cognition if you were assigned to the active treatment group on this trial across a 48-week study period. And the mean improvement in those who received AVXL 273 and who did improve, they had a mean ADAS-Cog improvement of about 4 points, which is clinically as well as statistically significant. In the ADCS-ADL, if you maintained or improved function over the 48-week study period, you're 167% more likely to have improved if you were assigned to the active treatment arm of this study. We haven't passed the results between the 50 and 30 milligram dose study arms at this stage, but in the 2A study, there was a clear advantage towards those who could tolerate higher doses. In the ADAS-Cog, we showed a slowing in cognitive decline by 45% compared to placebo at the 48-week time point, and the mean difference in ADAS-Cog score change was 1.85 points in favour of active drug compared to placebo. In the secondary endpoint using the CDR sum of boxes, decline was 27% less across both treatment arms than it was for the placebo arm, with a mean treatment difference in score of 0.42 points on the CDR sum of boxes, again with a significant p-value associated with that. In terms of safety, there was a design built into the study whereby, despite patients being randomised to placebo 30 or 50 milligram arms, the investigators at each site had the ability to up titrate or down titrate within the study period according to tolerability of the drug. We found that a large proportion of the patients on both treatment arms did tolerate up titration to the target dose, with significant numbers not experiencing a down titration episode during the maintenance phase of the study. In terms of adverse events and tolerability, there are a high rate of adverse events in any Alzheimer's disease population, both across placebo and intervention groups. You can see that the broad rates of treatment emergent adverse events between placebo and both active treatment arms was roughly similar. There were higher rates of serious treatment emergent adverse events in the intervention arms, however. We'll come to that. And there were 2 deaths during the course of the study as well, one in the placebo arm and one in one of the treatment arms. These were both felt to be unrelated to treatment. The first death in the placebo group was due to progression of the underlying disease, a patient with the posterior cortical atrophy variant of Alzheimer's disease, and the patient who passed in the active arm died from a urinary tract infection that evolved into urosepsis. There were significant rates of certain adverse events that differentiated the treatment arms and placebo arm. In particular, rates of dizziness and the emergence of confusional states were more common in the treatment arms. I guess that's to be expected with a CNS active drug, and it shows a degree of target engagement there. But the adverse events that had a prevalence of 7.5% or more, being dizziness and confusional state, were predominantly mild and moderate in severity and were managed by down titration. Also in this study, we used morning dosing for the patients in all treatment arms, and for future studies we'll be likely to change to nocte dosing to minimise the emergence in particular of dizziness as a treatment emergent adverse events. Confusional state, perhaps you might think it's surprising, but a number of patients who are prescribed cholinesterase inhibitors will also become confused. So it's not necessarily a flaw that's unique to this treatment drug by any means. So the safety findings in this study were consistent with what we'd observed from a phase 2A study, and not particularly out of the ordinary for a CNS drug in this particular population. There are a number of patients who did make use of the up titration and down titration ability that's retained within the protocol. The dose was changed in a significant number of active treatment patients compared to those who received placebo, but the number of drug withdrawals during the maintenance phase of the study was less than 6% in the treatment arms. And you can see the main reasons for drug being withdrawn during this maintenance phase were the occurrence of dizziness in 3.7% of the treatment population and confusional states in 2% of both treatment arms as well. In summary, we have a drug that seems to actively engage with the sigma-1 receptor, and through a fairly large-scale phase 2b/3 interventional study controlled by placebo, we're pleased to have shown a significant decrease in both cognitive decline at 48 weeks and a decrease in levels of functional impairment as measured by the ADCS-ADL, along with a significant decrease in worsening on the CDR sum of boxes. Again, very top-line results. I apologise for the lack of detail, but it's a function of the time within which we were able to get some useful data following database lock as recently as 2 weeks ago. Thank you for your attention, and we'll look forward to seeing further development of this drug. The company plans to publish a full analysis of all of the variables that were accounted for in the study, including whole genome analysis, analyses that are specific to possession of the sigma-1 wild type versus the mutation, as well as biomarkers on neurodegeneration. And there's a forthcoming publication in a peer-reviewed journal at which those data will be revealed. It's important to note that people who completed the double-blind study were eligible to roll over into an open label extension phase called Attention AD. That continues and will follow up patients who completed the double-blind phase for a total of some 96 weeks. And the company plans to meet with regulatory authorities to determine the next steps in the development of this potentially important compound. I'd like to end by acknowledging the contributions of the principal investigators at each site, as well as the study site staff, the Data Safety Review Committee, the AVXL Scientific Advisory Board, and of course, most of all, to those patients and carers who gave up their time, effort and energy to participate in clinical trials such as this and to produce meaningful results. Thank you for your attention.
We have time for one question.
I was wondering if there was a statistically significant difference between the ADAS-Cog scores between placebo and drug groups at the end of the study.
I think one of the slides spoke to that. There was a statistically significant difference of about 45% from memory between the aggregate treatment arms, whether they were 30 or 50 milligrams. It's not been passed down to that extent yet, and those who received placebo. So yes, there was.
Okay, thank you.
Can I have one question? Frank Weber, Zurich Life Science Consultants. You had a one-to-one randomized study, 3 arms, but then you pooled the 2 arms against placebo. And was that your predefined primary analysis, or the p-values you showed was always the pooled against the placebo, or did you just have one arm against placebo defined as primary?
Thank you for the question. The plan remains to analyse both treatment arms against placebo separately. The only reason we presented aggregate data today is the limitations of the timeline in getting the data set to AVX. As I say, database lock was 2 weeks ago. AVX received the data yesterday, and I received this slide set within the last hour. So that will all come in the fullness of time.
Thank you.